Welcome to this comprehensive tutorial on CGAS, the Chloroplast Genome Analysis Suite. In this video, we will walk through all 14 modules of CGAS, from installation to complete phylogenetic analysis. Whether you are assembling your first chloroplast genome or conducting comparative genomics across dozens of species, this guide will show you everything you need to know. Let's start by understanding what CGAS is. CGAS, which stands for Chloroplast Genome Analysis Suite, is a comprehensive Python-based toolkit that automates the entire workflow of chloroplast genome analysis. This is a complete pipeline that takes you from raw sequencing read all the way to publication-ready phylogenetic trees. The suite includes 14 specialized modules that cover every aspect of chloroplast genome analysis, including assembly, annotation, and comparative analysis. One of the key advantages of CGAS is its automated processing approach, which requires minimal manual intervention. This means you can process dozens of genome efficiently without tedious manual steps. CGIS is cross-platform compatibility, working seamlessly on Linux, macOS, and Windows operating system, and importantly, all output are publication ready, including Excel tables, Word documents, and high-quality figures. This makes CGAS perfect for researchers working with chloroplast genomes and plant genomics, phylogenetics, and evolutionary biology. CGAS offers two installation methods. Option 1 is CGAS complete. This is a full Conda environment that includes all dependencies. It is ideal for servers and high-performance systems where you have plenty of resources. CGI complete support all 14 modules and include everything mean, FASTP, get organally, BLAST, plus, IQ tree, and R packages. This is the recommended option if you plan to run the complete workflow from raw reads to phylogenetics tree. Option 2 is CGA's minimal. This is a lightweight setup perfect for laptop and standard desktop computer. It includes only the core dependencies and support modules 2 through 14. This means you can run all the comparative analysis and phylogenetics modules. However, Module 1, which handles genome assembly, requires some external tool to be installed separately if you choose the minimal installation. This option is perfect if you already have assembled genome or if you are working on system with limited resources. The first installation step is the same for both complete and minimal installation. However, I will show you just the process of installation for complete CGAS. First of all, we will clone the Git repository and run in the terminal this command. Then we will go to the CGAS directory. Now, what this does is download the complete CGAS repository from GitHub and create a folder named CGAS containing all the modules. The second command simply change your current directory into the CGAS folder so you are ready for the next installation step. Now, how we can specifically install the complete CGAS. So, we will just run this command in the terminal. When this completed, then we will activate the CGAS environment and then we will run just this command. And after running this command, we will check if the CGA is installed successfully or not. So this will install everything, including Git organelle, fastp, same tools, BWA, and the R packages, IQ tree. So we can use this for all the modules. But for the PGA, we need a separate installation that we will discuss in a separate video later, but in the README file you can find all the details. Now we will do practically the installation process, so please give a look to the complete installation process in the command line program and see practically that how we can install the CGAS. Now let's dive into the modules themselves, starting with Module 1, Genome Assembly and Quality Control. The purpose of Module 1 is to process raw sequencing read through quality control, assembly, and validation to produce complete chloroplast genome. This is the foundation of your entire analysis pipeline. This is the main command. Now, what this mean? So, let me explain. This command line tells the CGAS where to find the raw sequencing read in FASTQ format. This line of command show that where CGAS need to create output folder with all assembly result and QC reports. I will show you that how many subkit directories are formed and how the different results are present in these directories. The modules automatically find single in or fear in reads and process all samples. The final output will be complete chloroplast assemblies which you will find in the 07 assembled genome subdirectory. These assembled genomes are ready to move on to annotations in module 2. Module 1, I will show you the raw reads. Here, these are the raw reads, and we just need these raw reads. Just for module 1, we need this. After the completion, this will provide us the complete detail. We will give a look to the results of module 1. Just see, this is the directory. This includes the QC report. We can just give a look to just one sample. Just see, this is the report. And here we can see that the quality of all reads. Now, give a look to the second directory. This is the clean reads. Here, For each sample, the clean reads were generated, and these clean reads were used for the de novo assembly of chloroplast genome. This is the third one. Here, all the assemblies are present which were generated by Git organelle. These are the two contigs which are just different based on the ACC flip-flop orientation. All the results are similar to those which are specifically generated by the Git organelle. Yes, here we are just using the integration of Git organelle, and this is important to cite Git organelle. Specifically for QC report, we need to cite the first P. Here is the mapping report. Just see these are the BAME file which are generated. So we need to cite the BWA and same tool. Here the chloroplast specific reads are present. Just see. And these are the reports. We can specifically give a look to this one for further clarity, and we can see, just see here, we can see that the complete circular genome is generated by the Git organelle. Here the number of contigs are mentioned 1. As I mentioned to you that these two contigs are just different based on the SST orientation. This is the Q20 and Q30. This is the number of bases after QC in giga bases, and this is total number of reads after QC, and this is read before QC. So these are the complete detail. This is the mean coverage and this is the median coverage of the chloroplast genome. This show the mapping rate. So this provides you the complete detail and cover each aspect of the chloroplast genome assembly process. So this module 1 not only make the whole process automatic and integrating many softwares together, but also make the whole process efficient by generating the complete results in one single file, which can even be used directly in the manuscript. Here are the assembled genome. So we can see there is two contigs for each one. But as I mentioned, that these contigs are just different based on the orientation. So there we just choose to mention one contig instead of two contig because the size of the LSC, SSC and IRs are completely similar except the orientation of the SSC. And this gives you the complete report that how the samples are run, how many samples were found, how the process is completed. You can find this report in the fixture. Module to handle plastome annotation using PGA, the Plasted Genome Annotator. This module annotates your assembled chloroplast genomes, and the result is a high quality gene bank file with properly annotated genes, transfer RNA, ribosomal RNAs, and coding region. These annotated files are ready for comparative analysis. The main command has several components. This is the main command. Now I will explain that how each part work. This part, this part specify your target genomes to annotate. These are the genomes you got from module 1. This part, this part of the command provide a reference genome that serves as a template for annotation. This helps PGA predict gene more accurately. This part, this part that is optional. It is a text file that maps your sample name to scientific species name. If you don't provide this file, CGAS will use the FASTA header as organism name, which works fine in most cases. If you want to change the names of the organism later in the gene bank file. The output consists of gene frame file for each genome complete with gene transfer RNA, ribosomal RNA, and structural feature. These gene banks files are the format required for all downstream comparative analysis modules, but you will need to do some curations for the correction of annotations, which is important step, and some manual work is required. Run here. But first I want to show you that how the files are present. Just see, this is the reference genome here. We place just one reference genome in this folder. In this folder, we have given the genomes which we need to annotate. I have given these files to annotate. And here we provided the organism names. Just see here. Now we will give the command. Here, how we can give the command, just like first we need to activate the conda. When the conda become activated, sorry, conda activate CGAS. Now we need to provide the command. Just see, this is running. This will complete just in few minutes. If you don't want to provide this command, then still this will work. You can look to the README file for setting up the PGA. Now I will show you the result after completion of this step so you can find out that how the annotations can be performed. Just see here. If we come to this one again, then we can find out here. Just see first this is completed and this gave us the complete result. The total file process 4 successfully annotated 4. Now look here to the annotated GenBank file and we can see all of these because just see here the header was just changed based on the 1 and 2 while the name of the organism was similar. So we can see this one, but in our file, this one was like this. So the script automatically take this one just for one sample and this one for another sample because these two genomes were just different based on the SEC orientation. Just see here, if we open this one in the TextPad file, you can find out that this provides you complete result. Usually PGA just provides you the result of the IR regions, but this one will provide you the complete results of the LSC region, also the SSC region, and also the repetitive region, mean inverted repeat A and inverted repeat B. This will provide you the complete result, and also this will include the species name. So you need, if I will suggest that you follow the whole command line to save time in the future and in the downstream analysis. Module 3 is about standardizing gene name across all the genomes and detect missing gene. So therefore we specifically name it gene normalization and comparison. Why we are using this module? Different annotation tools and different annotator often use different naming convention for the same gene. Module 3 detect missing gene, extra gene, and annotation inconsistencies. The output ensure that all your genome follow a consistent naming system, which is absolutely essential for accurate comparative genomics. The command is straightforward. Here, this part, reference genome.gb part, this parameter specifies your reference gene bank file for gene name standardization. This should be a well-annotated genome from a closely related species. Here, how it's work. All other gene bank files in your current directory are treated as target genomes. The module normalizes their gene name to match the reference naming convention and then create a comparison report showing gene differences across all your genome. And then you can also compare that what is the size of the CGAS and what is the product name of the different species, and you can also find out that in which genome or in which species the specific genes are mentioned with the intron, but in which species the genes are not mentioned with the intron. So if a gene is mentioned as intron containing gene in one species, but mentioned is a gene which does not contain intron. So this clearly indicates if this is either related to the annotation error or there may be a real loss of intron. Now I will show you that how this works. Regan agrees is the reference. Now all the other genomes here will be considered as target genomes. I also want to show you here. Here we can see that all the genomes having space, but this one is not having any space. This one is using as a reference for all these genomes. From now on, I will try to use the time stamp. So just see here, this just include in seconds. And in second, this provided us the results. Just see here. I specifically included such kind of naming inconsistencies. And we can see that our script can find out these inconsistencies in the genome. Now we will give a look to the results. So here are the revised annotations. All these gene banks are the newly generated gene banks, and these contain the genes with the name after normalization. And here are the result for the comparison, and you can see that this is the genome, and this genome contains the CDS, and is this present? This is true. And then for the intron, we specifically mention is the reference genome contain intron, is the target genome contain intron. So for both this is false. If one is true and another was false, then this will be very technical and we will need to check again this part very carefully, and we will need to check that is this is here is a gain or loss of intron, and is that is real one or this is just related to the annotation problem. So the same we can find out the variation in the length of the CDS or in the length of the intron, something like this. So this is very important and give us a lot of information. Module 4 Format Conversion and Validation. Module 4 handles format conversion and validation, which is crucial if you plan to submit your genomes to NCBI. This module validates annotation and prepare files for NCBI submission. It generates submission-ready file and check for annotation error, improving data quality before publication or database submission. The command is very simple and could not be simpler further. You just need to write the CGAS module 4 and this will work. The same pattern is followed by the module 5 to module 13. How all these modules work? This not require any input file. This will process all gene banks files or the FASTA file, but in case of module 4, the gene bank files in the current directory. This will convert gene bank file to FASTA format and also generate the .tbl format. This will validate gene location and annotation quality, auto-correct common annotation errors, but in some cases we also need to do manual curation, create individual and combine submission files. Just see, this even completed for the 10 genome, this complete in seconds. Here is the file. We can see combined FASTA file. Just see this file is created for submission to NCBI. Similarly, the feature tables are generated for submission to NCBI. Now give a look to the validation. So in the validation, we can see that the PSBL start with the ACG. So this may be correct because ACG, as we know, can be converted to AUG by the RNA editing. Similarly, RPS19 having GTG, and this is also a common start codon for the RPS19. But if for some other gene this is, or if some other start codon is present, or there is internal stop codon, or the terminal stop codon is not exist, or if there is missing of the CDS, or if there is the issue with the transfer RNA naming, then the script will indicate those issues and then manual curation will be needed for the correction. But this shows that all the genome which we are analyzing are perfect for the annotation so far. Now we will give a look to module 5 to module 13 first here, and then we will run all the modules together. So first we will just check that how all these modules work. The module 5 is for gene comparative analysis. This module compares gene content across species. It highlights gene presence, absence, duplication, and variation pattern among genomes. The command is very simple, just CGAS module 5. What this means? This process all gene bank files in current directory. Extract all genes, CGS, transfer RNA, ribosomal RNA from each genome. Normalize gene names across different annotation convention. Here we again included the option. Classify genes as functional or pseudogene. Detect IR-mediated gene duplication. Create gene presence and absence matrix across all species. Module 6 is for gene content table. This generate publication ready table in Word format. These tables summarize gene distribution and can be directly included in the manuscript. Just run with the CGAS module 6. This means all the gene bank files present in our directory will be used as target files and the Word document will be generated. Those will include the gene names by functional categories. Table will be ready to include in the research paper. Module 7 is for genome structure analysis. This examines LSC, SSC, and IR region. It calculates genome size, region boundaries, and GC content, helping interpret structure variation among species. The main command is just CGAS module 7. This also processes all gene banks files in current directory, identify LSC region, SSC region, IR regions, and calculate DC content for each region and overall genome, and also for the functional region like coding region, transport RNA, and ribosomal RNA. Module 8 is for codon usage analysis. It evaluates relative synonymous codon usage. It identifies codon bias pattern and provides visualization for evolutionary interpretation. The main command is just CGAS module 8. What this means? This process all gene banks file in current directory, extract coding sequences from all genome, calculate RSCU value for each codon, compare codon usage pattern across species, generate R-based visualizations. Module 9 is amino acid analysis. This calculates amino acid composition across species. This helps detect conserved and variable protein level pattern. The main command is just CGAS module 9. This process all gene bank file in current directory, translate coding sequences to amino acid, calculate frequencies and percentage of each amino acid, compare amino acid composition pattern across genome, generate R-based comparative visualization, identify conserved versus variable protein characteristic. Module 10 SNP analysis. The purpose of this module is to identify nucleotide substitution across genome. It provides substitution statistic and visual summary of sequence variation. The main command is CGAS module 10. This process all FASTA alignment in current directory, calculate substitution frequencies and types, generate quality visualization showing SNP distribution, useful for phylogenetics and marker development. Module 11 Intron analysis. This compares intron containing gene across species. It detects intron presence, absence, and structural differences. CGAS Module 10 is the main command. This process all gene bank file in current directory, identify genes containing intron, both protein coding gene and transfer RNA genes. Compare intron presence or absence pattern across species, calculate intron length and count it. Create complete comparison table across all genome, reveal evolutionary pattern of gene structure. Module 12 SSR analysis. This identifies simple sequence repeat. It classifies them by genomic region and motif type, generating high quality visual output. The main command is CGAS module 12. This process all gene bank file in current directory, detect microsatellite repeats, mono, di, tri, tetra, penta, and hexanucleotide repeat. Map SSR to genomic region, LSC, SSC, and IR. Assign SSR to functional region, CDS, and tran intergenic spacer. Compare motif type and distribution across species. Generate R-based publication quality visualization. Module 13 Nucleotide Diversity Analysis. Purpose: calculate nucleotide diversity value. It identifies high variable region that may serve as molecular marker or DNA barcode. The main command is CGAS module 13. This process all gene bank files in current directory, compare all regions of the chloroplast genome, calculate phi value for each region, identify high variable regions suitable for barcoding, and also identify conserved region under selection pressure. Generate the R-based high-quality visualization plot. Now we will run these modules together. This is very simple. We will just mention time and we will just mention here modules like 5, 6, 7, 8, 9, 10, 11, 12, and 13. Just see, all these modules will be completed one by one. Now, some of the modules will be already generating the results, so we will just look to the results and will explain the result in the meanwhile. This one is just see module 5, so we can give a look here. Just see here, this provides us the complete result that this gene is present just as a single copy. This gene is just present as a functional copy, and this have two functional copy. One is in IRA and another one is in IRB. Similarly, this provides us all the information for the genes. Just see here, RPS19, here is two copy, and this provides us the clear information that one is functional copy and one is pseudogene copy. Now look to the summary. In the summary, this provides us very clear information that all the genes having similar gene and the duplicate genes are also similar. This also mentioned the duplicate gene here. Genome specific gene, there will be no genome specific gene because all the genes are similar. Missing gene, so we can say here that there is no missing gene so far. Just see, in all the species, these genes are present. If there is difference in the number of genes, then we can identify that the number of genomes are 10 and present and also in 10. So this means all the genes are similar in all the species. This also mentions the genome and the species name. Missing from genome, so from none. So this is very important. This provides us complete detail that if there is any gene which is missing In a specific species, this will give us the information. This also do some normalization, but this is not like the module 3. Just see, this is the specific option which I included for the normalization. Now look to another one. Here we can see that this provides us the complete table. This is the separate table of each one, but we will look to the combined one. Now we will look to the combined one, and we can see that these genes are present here in the genome. 130 gene. This also mentioned the C2 genes to us, and this also mentioned to us the genes with two intron and single intron like this. For the RP gene, they mention very clearly that the RP gene is a transfly gene with two introns. So this is very important results. Now give a look to this one. Just see this provides us the complete detail. Just give a look to this. This provides the length of the complete genome of the LSC, of the ACC and IR region. This also provides the information of the GC content of each region, and this also provides the GC content of the transfer RNA, ribosomal RNA encoding sequences. Now give a look to this one. This provides us the complete detail here for each one, for all the species in the table form, and also this provides us the figure. Just see here in the PDF format and also in the PNG format. Just give a look to this one. Just see here. This provides us very clear information. Similarly, for the amino acid, we can see here. Just see here, this one completed. So I want to just show this first. This take just 4 minutes and 56 seconds to complete all the modules from modules 5 to module 13. So this show that these are the actual script which I prepared for the publication purpose. So this show that how efficient is the whole process using the CGAS software. Now give a look to this one. This gave us the information about the heat wave, and you can also see the result like for the three representative genomes. And here you can see this one. This provides us the data in the form of table. Now we need to give a look to the module 10. Just look to the figure first. Just see, this is in the horizontal direction, this is in the vertical form. This is for the transition and transversion ratio. And you can also give a look to the table. So in the table form, this provides us very clear information. Here is the intron data. This provides us the intron information that from where the intron start and where the intron end and what is the total length of the intron. Similarly, this gave us for the transfer RNA. Then this also gave us the comparison that in which species the intron size was minimum and in which species the intron size was maximum and what was the difference and what was the mean size of the intron and in how many species the intron was present. Similarly, for the transfer RNA gene. Now give a look to the SSR. For the SSR, we can see the detail. You can give a look to other detail later, but I just want to show you. Just look here. This gave us the complete information that where is the SSR, what is the location of the SSR in the genomic region, what is the location in the functional region, and is either IGS, intronic, or the protein coding gene. Just look here. And this also provides the complete annotations and abbreviation detail for the publication ready table. Here we can see the figure. Just give a look to the figure. Just see these are the details. Here we can also see the summary. And in the summary, everything is provided. Just give a look. Now give look to the diversity. For the diversity, we will directly give a look to the figure. Just see, this is the figure draw and the region were identified with the nucleotide diversity. This is the two panel. One is for the coding region, one is for the non-coding region. Now we will look to the module 14. The module 14 is related to the phylogeny. The module 14 uses the IQ tree. So the purpose of module 14 is to build concatenated gene matrices and reconstruct phylogenetic tree using IQ tree. So here we mentioned the gene matrices. So the gene matrices are specifically using for the deep divergence. But if someone want to give a look to the population genetic level or the very closely related species of the genus, then there is the option to generate the matrices from the intergenic spaces region and tronic region and also from the complete chloroplast genome. You can either use the marks aligner or you can use the mapped. If you didn't specify the marks, then the mapped will work automatically. If you want to work with the marks, then this will use the marks. So the final output will be publication ready phylogenetic tree. This means that you will get the final version and then you will need to draw the phylogeny in the factory or in the ITOL. What this command mean? Here the mask is for the aligner. If you didn't mention, then the map will be used automatically. This gene only is to use only protein coding gene for the phylogeny. IQ3 mean that run IQ3 for maximum likelihood phylogenetic inference. OG mean outgrowth. So this means specify outgrowth species. So we will need to mention a gene bank file. And module extract gene, align them, concatenate and build tree. So this module is very, very efficient. So I will want to show you that how this work. If this is the command. So, okay, we will write it by ourselves. Like this will first align all the genes and then this will draw the phylogeny. Now, this provides us all the output files and generated the tree. This gave us that which parameters are used, and this also mentioned that B in an I option was enabled and the threads were auto. This also gave us the information that the partition file was gene only and executable were IQ tree. Here I didn't use the timestamp because the IQ tree is taking a specific time and this is not dependent that we run whether using the CGAS or use the manually with IQ tree. Finally, thank you very much for watching this tutorial. Hopefully you will find this helpful for your research.